I'm very honored to introduce today's lecturer, Dr. Michael Gottesman. Dr. Gottesman has been Deputy Director for the Intramural Research at the NIH since 1993. He is a graduate of Harvard College and Harvard Medical School. Dr. Gottesman completed his residency at Brigham Hospital in Boston. He was a research associate at the NIH from 1971 to 74. He then returned to Harvard Medical School as assistant professor before returning to the NIH in 1976. Dr. Gottesman became the chief of the Laboratory of Cellular Biology in the National Cancer Institute in 1990. During his 26 years of service in the U.S. Public Health Service as a commissioned officer, he achieved the rank of two-star Rear Admiral as Assistant Surgeon General. His research interest has mainly focused on how cancer eludes chemotherapy. He's published extensively on this subject with over 500 scientific publications. Dr. Gottesman is elected fellow of AAAS and the American Association of Physicians, and a member of the National Academy of Medicine and the American Academy of Arts and Science. I'm sure you will enjoy today's lecture.
My name is Michael Gottesman. I'm the chief of the Laboratory of Cell Biology at the Center for Cancer Research at NCI at NIH. And my talk today is about the structure and function of ABC transporters in health and disease. This work began more than 30 years ago as an effort to understand why it was that some cancer cells were resistant to anti-cancer drugs, whereas others responded well to chemotherapy, and led, as you will see, to the discovery of transporters that are involved not only in drug resistance in cancer, but in handling drugs, many drugs, in different compartments in the body as well. So our overall goals when we began this work, and in current studies as well, were to define the molecular mechanisms of drug resistance in cancer. And we focused mostly on natural products. These are drugs that are traditionally used to treat cancer and to which many cancers respond quite effectively. And also compounds which include platinum, such as cisplatin, which are also effective anti-cancer drugs. We discovered in the course of our studies a number of different mechanisms of resistance to these agents, and we've been engaged in trying to determine the clinical relevance of mechanisms derived from these in vitro studies to actually affect resistance in patients' tumors. This work has helped us develop new approaches to exploit or circumvent clinically significant resistance mechanisms, and has led to a better understanding about the cellular pharmacology and the pharmacokinetics of drugs in the human body. Now, the issue of drug resistance in cancer can be summarized in this slide. And that is, for most metastatic cancers, those are cancers that have spread from one part of the body to another, chemotherapy with classical or targeted anti-cancer drugs can result in remissions, but frequently does not cure the cancer. And this is shown schematically here as a population of cells that may be intrinsically resistant and so not responsive to anti-cancer drugs at all, or essentially sensitive but containing some resistant drugs, which over the course of therapy become the major population within the tumor, and so the tumor acquires resistance. There are two major hypotheses to explain the development of multidrug resistance, which can result in treatment failure. The first is that within the initial population of tumor cells, there are a few cells, perhaps a small subpopulation, that are already resistant to the therapy which is going to be employed. Evidence exists in acute myelogenous leukemia and in melanoma that that is indeed the case, and that when you treat a tumor, you get remissions, and the cells that grow back were originally present in the original tumor, and that can be demonstrated using sophisticated molecular techniques. The second basic model is that there may be resistant and sensitive cells in the initial population, but that during the course of therapy, additional mechanisms of resistance develop, and that these may become fixed during the course of therapy, leading to mixed resistant populations. Although these seem to be complementary mechanisms, the approach to dealing with this kind of resistance is quite different. In the first case, you want to derive a therapy which kills as many of the different kinds of resistant cells as possible in the first treatment. Whereas in the second case, it's really not known what kinds of mechanisms of resistance will derive, and therapy needs to be personalized for each tumor so that you can identify which mechanisms may be present in an individual patient's tumor. This, of course, makes therapy much more difficult, and in many tumors, this appears to be a major way in which resistance develops. Our goal was to understand at the molecular level what mechanisms might pertain in each of these cases. So let me take a step back before I go into some details and describe some of the specific mechanisms that occur that cause resistance in tumor cells. For therapies that are targeted, that are aimed at specific oncogenes that may be responsible for tumors, we often see mutations in these targets or bypass mutations. For example, if the target is a growth factor receptor and the drug no longer inhibits that specific target, you may see alternative ways of activating the tumor using other kinds of pathways other than the pathway that is being targeted. A second mechanism shown to the right here is that altered cells may show altered cellular pharmacology. And this is a mechanism we've been studying in considerable detail and has enabled us to develop an understanding of how drugs are handled in the body based on how cancer cells handle these drugs. And this includes mechanisms that may reduce the accumulation of drugs or alter metabolism of drugs or increase efflux of drugs from cells. A third change, which turns out to be rather important in development of drug resistance, is changes in differentiation pathways and homeostatic responses within tumors. So a tumor that begins as an epithelial kind of tumor can change into a more mesenchymal tumor with massive changes in gene expression, resulting in very different patterns of resistance to anti-cancer drugs. And the classic example is what's called epithelial to mesenchymal transition. And finally, there are important alterations in the local environment of tumor cells, including the presence of other cell types that are not cancers but can affect response to chemotherapy, changes in the substrate in which the cells are sitting, including changes in tissue plasticity and changes in elasticity, and alterations in mechanisms including immune mechanisms and mechanisms that affect growth of blood vessels into tumors. All of these undoubtedly contribute to drug resistance, but our effort has been focused on those that change cellular pharmacology. Now, if you look at cell-based mechanisms of resistance, there are three general classes of alterations in the tumor that can result in reduced efficacy of chemotherapy. The first and most obvious is that the drugs simply don't get into the cells. We know there are close to 400 different solute carriers which are responsible for moving normal nutrients and other agents within cells. And approximately 10% of them have been demonstrated to also affect uptake of specific drugs into cells. And reduction in amount or changes in the specificity of those transporters can produce drug-resistant cells. If the drugs can get into the cells, there are mechanisms that affect cell biology which have profound effects on drug resistance, such as reduced cell killing by cell-killing pathways known as apoptosis, altered cell cycle checkpoints or growth pathways, altered metabolism of drugs within the cells, altered targets or increased repair of damage, and even compartmentalization of drugs within subcellular compartments that prevent them from reaching their targets. Now, much to the surprise, I think, of many people in this field, it was discovered approximately now 35 to 40 years ago that a major mechanism of resistance is actually related to the increased energy-dependent efflux of drugs from cells. And we were involved in cloning some of the original transporters that were responsible for efflux of drugs. And it was soon discovered that these belong to a family of ATP-dependent transporters called ABC transporters, of which 48 are known in the human. Now, these transporters turned out to be particularly interesting, both in terms of their biology, their biophysics, and in their cellular pharmacology. We've shown that they play an important role in multidrug resistance in cancer. And this is true also in normal cellular pathogens, including protozoans and bacteria. These kind of transport systems turn out to be rather important. They play a role in pharmacokinetics, that is the uptake, distribution, and excretion of drugs in the body. They are important in drug toxicity because tissues that have efflux pumps are less likely to be affected by drug toxicity. One example that I'll mention, for example, is the stem cells in the blood, in which case the expression of these transporters makes stem cells somewhat more resistant to anti-cancer drugs than you would expect otherwise. They play a key role in development. I mentioned their expression in stem cells. They're involved in morphogenesis because they can keep normal products from entering cells. And we've learned that they are important in understanding the biology of all transport systems. So let me focus now on the ATP-binding cassette or ABC transporter superfamily. It is one of the largest families of transport proteins known. Currently, more than 2,000 members have been identified. Substrates include many different kinds of small molecules: ions, sugars, glycans, phospholipids, cholesterol, peptides, proteins, toxins, antibiotics, and, as I'll soon show you, hydrophobic natural product anti-cancer drugs. Structurally, they consist of various combinations of ATP-binding domains and segments that include transmembrane domains. The family of eukaryotic ABC transporters includes 57 different known genes, and this shows the evolutionary profile of these different genes. Mike Dean, a senior investigator at NCI, has really elucidated these various families. And you can see that the human transporters are closely related to those in primates, but also to chicken and other mammals and reptiles as well. Every single known family of living organisms includes representatives from this family. Now, in the human, as I mentioned, there are 48 human ABC genes. They fall into 7 different families. And the families are defined by their sequence homology, but also by their structural homology. So, for example, in the ABCA family, there are 12 members, and all of them, as shown at the bottom right, consist of 2 segments, each of which contain 6 transmembrane domains and 2 ATP-binding cassettes. For the ABCB family, which includes several members of the family that includes multidrug resistance proteins, you can see that, again, we have 6 transmembrane domains and 2 ATP binding cassettes. But there are members of this family, as for the ABCG family, that include only a single segment that has transmembrane domains and 1 ATP binding cassette. And evidence suggests that these form dimers, so that the coin of the realm here are the 2 6 transmembrane domain segments and the 2 ATP binding cassettes. Now, the transporters are involved in many different physiological processes. There are 3 that seem to have a very wide specificity, are multidrug transporters, and those are known as ABCB1, generally called P-Glycoprotein or PGP. This was the first of the family to be discovered in the 1980s, cloned in the 1980s, and led to the discovery of the other transporters. ABCC1 or MRP1, multidrug resistance related protein, is also expressed in many different tissues, as is ABCG2, also known as BCRP, standing for breast cancer resistance protein, and mitoxantrone resistance protein. And many different laboratories have been involved in identifying, cloning these transporters, and understanding their drug specificity. If you look to the right, you can see a list of different drugs that are substrates for P-Glycoprotein. Since P-Glycoprotein sits in the plasma membrane, these drugs are unable to accumulate in cells that express high levels of P-Glycoprotein. And the tissues that express this protein are shown in the lower left column, and I will describe in a moment in a little more detail how these affect the distribution of drugs within the body. Now, it's been demonstrated that there are a number of different human diseases where expression of transporters affects the development of the disease, and in most cases these are simple Mendelian dominant disorders. I mentioned cancer and the multidrug resistance genes, but for example, cystic fibrosis, the gene that is responsible for this very common genetic disorder which leads to lung disease in human populations, is the transporter ABCC7. In this case, the primary function of this transport system is as a chloride channel, but it's a regulated chloride channel, and the ABC-binding cassette is involved in opening and closing of the channel. And then you can see from this list that there are a variety of other diseases, and although we do not know diseases associated with all 48 of the transporters, this list continues to grow, and it looks as if most of them subserve specific functions. When they are absent, the effect is not lethal, but it does result in disease which can be quite debilitating for human beings who have these problems. So to get back to those three multi-drug transporters, ABCB1, ABCC1, and ABCG2, their overall structure is shown here. As you can see, as I mentioned, for ABCB1, there are two regions which have 6 transmembrane domains, and there are 2 ATP-binding cassettes. For ABCC1, there's an extension at the N-terminus of 5 transmembrane domains. Evidence exists that that can be deleted, and the ABCC1 is still a multi-drug transporter, and it's thought to be involved perhaps in localization of the protein or stabilization of the protein in the plasma membrane. As I mentioned, ABCG2, another multi-drug transporter, which shows only one segment of transmembrane domains, appears to exist in most cases as a dimer, fulfilling the requirement that you see for ABCB1. Now, in the human, these 3 multi-drug transporters have some overlapping drug specificity. This is shown in this Venn diagram. If you look in the middle, you can see a variety of different anti-cancer drugs, including both targeted anti-cancer drugs and more classical drugs that are not specifically toxic but do kill cancer cells. These are very effective anti-cancer drugs, but in the presence of any one of these transporters, their efficacy is reduced. You can also see that there are specific agents, including anti-cancer drugs and other intermediate metabolites, that are affected by one or the other of these specific transporters. Now, this is the situation in the human. Most of our studies on understanding the pharmacology of drugs and the effect of these transporters is done in rodent systems, either the mouse or the rat. And in those cases, the specificity of each of these transporters is slightly different. So being able to extrapolate from what we learn in the rodent systems to human is not necessarily totally straightforward. In many cases, the specificity is similar, but in some cases, it actually differs. So let me get back to this issue of what the normal function of P-glycoprotein is, and the information is provided mostly in localization studies. It sits, for example, in the small and large intestine on the luminal surface, so that if drugs are taken up orally, the ones that are substrate for PGP are unable to be absorbed, because as soon as they enter the plasma membrane of the intestinal epithelial cells, they're pumped back into the lumen and they are fecally excreted. The transporter is also present at high levels in the biliary epithelium, and so some of these drugs are excreted into the bile. It's in proximal tubule cells of the kidney, so drugs can appear in the urine. And it plays a really important role at the blood-brain barrier and in other barrier sites, such as the blood testis, the blood ovary, and the blood placental barrier, pumping material out of these specific organs to protect them. It also can be expressed in either tumor cells or circulating cells in the vascular space or the interstitial space, conferring resistance to anti-cancer drugs and insensitivity to other pharmacological agents for which the P-Glycoprotein is a transporter. So in our early studies, we asked what the relative specificity was of these different transporters for conferring resistance. We did these experiments in a couple of ways. One was to simply measure the level of transporter in different cells and find out what transporters were responsible for resistance to what agents. The other way was to select for resistance to specific drugs and ask where overexpression of transporters conferred resistance. And you can see that almost all of the standard anti-cancer drugs we can find at least one transporter that's responsible for creating resistance, either by selection or simply by measuring expression of high levels of a transporter that confer resistance to these drugs. So it's almost a truism that no matter what the agent that's used to treat cancer, one or more of these transport systems will effectively pump drugs out of the cells. That doesn't mean that all anti-cancer drugs express these transporters, but when they do, it's pretty clear that resistance will develop. So I'd like to focus a little more now on the biophysics and physiology and biochemistry of these transport systems. This is a linear diagram showing the 1,280 amino acids that make up human P-glycoprotein. This protein consists of two halves and a connecting region that connects these two halves. As you can see, there are phosphorylation sites in the region that connects the two halves of the protein. These are shown in red. We've deleted those sites and made it impossible to phosphorylate them. And it doesn't look as if there's much effect on the transport activity. We can't rule out some regulatory effects of phosphorylation, but phosphorylation is not essential for function. The yellow circles represent the regions, the parts of P-glycoprotein in which mutations have been shown to change drug specificity. These are clustered around the transmembrane regions. And as I'll show you in a moment, this led to the hypothesis that the place in the transporter in which the drugs interact is actually within the lipid bilayer. And that's also supported by photoaffinity labeling studies, which are shown in green here, in which transmembrane segments 3 to 6 and 9 to 12 are specifically photoaffinity labeled, consistent with the data from the genetic studies showing that these are important regions for drug binding within the transporter. So this led to a model that we proposed in about 1988, which specified that the transporter sat in the plasma membrane, that the site of recognition of the substrates was within the plasma membrane, and I just need to let you know that virtually all of the substrates for these anti-cancer transporters, anti-cancer drug transporters, are very hydrophobic, and in biophysical studies they prefer to partition into lipid bilayers compared to cytoplasm or extracellular space. And this model was proposed as I mentioned, quite some time ago, suggesting that the transporters were recognizing drugs within the lipid bilayer, and that we did know at that point that both ATP sites were essential for function. And we were interested in understanding the mechanism by which many different drugs could be recognized. And the binding of the drug led to activation of ATPase and the efflux of drug from the cells. Several years ago, it became possible to get crystallographic structures of P-glycoprotein and other ABC transporters. Steve Aller and Jeff Chang published now several years ago the structure of the mouse P-glycoprotein that's shown in the diagram on the left. This was not an extremely high resolution structure, but it demonstrated that substrate could actually bind to P-glycoprotein, and indeed was binding within the transmembrane regions in the lipid bilayer. In the structure that was published, the two ATP sites were very far apart, and evidence suggested that the active form of the protein in which ATP could be hydrolyzed, the ATP sites were close together. That's shown in a model on the right, which Kasper Locher published, looking at an ABC transporter from a bacterial species, Staph aureus. The model that we have is that the P-Glycoprotein exists in both states, and that when drug is bound, the two sites get together, and when they get together, ATP is hydrolyzed and drug can be extruded. Now, what is the evidence for this? So, my colleagues in the Laboratory of Cell Biology, Suresh Ambudkar and Sriram Subramanian, were able to develop with Sunit Shukla's support, we're able to develop cryo-EM structures. This is a technique for using a very high-resolution electron microscopy to study structures of different proteins. And as you can see in the upper left, we found roughly equal amounts of the open and the closed form of PGP. Based on the fact that in studies in which ATP hydrolysis could be blocked with vanadate, and we found only the closed form and therefore the active ATPase form of PGP, we've postulated that there's a cycle in which the open and the closed form are intermediates. Substrate can bind to either of these forms. When ATP binds, the only form that's present is the form in which the two ATP sites are close together. Once ATP is hydrolyzed, the P-glycoprotein changes its structure, releases the drug into the extracellular space, and then is free to recycle again. Now, the precise way in which substrates were recognized by the transporter was also quite mysterious. And Disha in our laboratory was able to generate roughly 9 different crystallographic structures of PGP. And he found that the two ATP sites were variable distances apart, in this series ranging from 45 angstroms to 60 angstroms. And that when he looked in detail at the transmembrane regions, the parts of the transmembrane regions that were exposed with the different distances between the ATP sites were different. So that as the two ATP sites moved together or apart, as they did under equilibrium conditions, different residues were exposed in the transmembrane regions, allowing the binding of many, many different substrates. So this led to the following hypothesis. The motion of the two nucleotide binding domains with respect to each other creates torque in the transmembrane domains that bind substrate. This allows PGP to sample a large number of potential substrate interaction sites in the transmembrane domains. And this, to some extent, may account for the enormous lack of specificity, or at least the specificity for many different drugs that are recognized by this transporter. The proof that this is actually the model will require much higher resolution crystallographic structures with different substrates bound to demonstrate that different substrates bind to different P-glycoproteins in which the ATP sites are at different distances from each other. And that as we change the distance between the two ATP sites, we change the substrate specificity of the transporter. And these studies are in progress. So I'd like to end by getting back to this issue of what the role of P-glycoprotein is in cancer. We believe that approximately 50% of human cancers express P-glycoprotein at levels sufficient to confer multidrug resistance. This has been shown in multiple studies. P-glycoprotein is commonly expressed in a lot of common cancers. For example, in tumors that have been selected for resistance to drugs, we find PGP expressed at high levels in leukemias, myeloma, lymphomas, breast and ovarian cancer. And that in situations in which we inhibit the expression of PGP, the sensitivity of these tumors to drugs can be reduced. We also see P-glycoprotein in cancers which express PGP at the time of diagnosis, including many solid tumors such as colon cancer, kidney, pancreas, and liver cancer. And it's generally known that these cancers don't respond to PGP inhibitors up front. So they are intrinsically resistant, and the tumors I mentioned before demonstrate acquired resistance. In animal models using human cancer xenografts, or models in which tumors are derived directly from the animals themselves, show that expression of PGP is a major mechanism of resistance, that inhibitors of PGP confer sensitivity. And this has led to an optimistic view that much of drug resistance might be reversible by PGP inhibitors. However, progress so far has been slow, although a number of different PGP inhibitors have been tested. In many cases, only transient response or no response at all has occurred, suggesting very strongly that although PGP may be sufficient for drug resistance, it's not necessary. It may coexist in many cases with other mechanisms of resistance. And so our search for all the different mechanisms that confer resistance goes on. But in the meantime, we've learned quite a lot about how drugs are handled in the body. In another lecture in this series, Matt Hall will talk specifically about the role of PGP, ABCG2 and ABCC1 in the blood-brain barrier and the blood-placental barrier. And I think you'll see at that point how the studies that I've described today are leading to a much better understanding of drug-drug interactions and the normal handling of drugs within the human body. So thank you for your attention, and at this point I will end my lecture.